This week we're going to be talking all about the muscular system. So you're going to have a few different functions of the muscular system. When we think of the muscular system, we primarily think of movement through contracting and relaxing those muscles to pull on our bones. Our muscular system is also important for providing support and posture, generating body heat, and helping us with things like our heartbeat, digestion, and blood pressure. We're going to talk about three different types of muscle tissue. We have cardiac tissue, which we find in the heart. We have smooth muscle tissue. We find that lining hollow organs. Hollow organs are going to be things like your bladder and your stomach and things that are made to fill up with stuff. And then also smooth muscle lines our blood vessels. We also have skeletal muscle. These are what we think of when we talk about muscles, right? Our skeletal muscles are going to be attached to the skeleton and the skin, and these are going to be our voluntary muscles that we're able to use to generate that movement and force. Muscle cells are known as muscle fibers, and muscle fibers contract and relax in order to cause movement. Here is another picture. Throughout this lecture, you'll see that I put a lot of just additional pictures in here and stuff for you. That is just so you can have a few different ways to look at things and to be able to use the study tools. So this is one of these. Here's your three different muscle types and what they look like under the microscope. The first type we're going to talk about is cardiac tissue. Cardiac tissue is going to be found in the heart. It is responsible for the rhythmic involuntary contraction of the heart. The cells or the muscle fibers are going to be striated. When we see that word striated, we know that that means striped looking. They're going to be long and branching. So you can see long, but also they're branching off. So they're not just in straight lines. And the way that we differentiate this under a microscope is we look for these gap junctions called intercalated disc. Intercalated disc are these little lines that you see running through them. The reason why those intercalated discs are important, and here's another view of these here. So more intercalated discs. The reason why intercalated discs are so important is because they make the cardiac muscles be able to communicate with each other better to cause really, really precise contractions of the muscle tissue. So whenever you're trying to contract an arm or a leg or something like that, it's not that big of a deal if the muscle coordination isn't super, super precise. Whenever you need your heart to beat, any like minute change in the way that that muscle tissue moves and coordinates together can cause really big issues for cardiac arrhythmias and things like that. So these gap junctions help all of the muscle tissues communicate so that they can all beat and contract together in exactly the way that they need to make the heart function correctly. We also have smooth muscle. This is going to be found in the walls and linings of the blood vessels and those hollow organs we talked about earlier, like your bladder, your stomach, intestines, things like that. These are going to be responsible for slow, sustained, and tireless contractions. So this very much, whereas your skeletal muscles are, rapid, they're strong, they're putting out a lot of force when they contract. These are definitely like the tortoise in the tortoise and hare race. So they're going for very slow contractions, but they're able to sustain them longer, and they're able to do this over and over and over again without tiring out because it's requiring a lot less energy to do these contractions. So when we're looking at these under the microscope, these fibers are going to have no stripes, so no striations. They're going to be long and smooth, and they can contract in multiple directions. Our last type we're going to look at is skeletal muscle. Like we talked about earlier, this is going to attach to the skeleton. It's going to be responsible for strong voluntary movement. But these muscles are going to tire out really, really easily. These cell fibers are going to run parallel to each other. They're going to be long, they're going to be striated, and they're going to be multinucleated. What multinucleated means is when we look at these cells, we see that they are going to have more than one nucleus per cell. So that's how to differentiate all of that under the microscope. So let's look a little bit more at skeletal muscle anatomy before we get into specifics of specific muscles and things like that. So the skeletal muscle is going to be composed of tightly packed bundles of muscle fiber cells held together by connective tissue. And I'm going to break this down a little bit more for you. So when we're looking at this as the biggest level, we're going to see the body of the muscle, right? That describes the whole muscle when we're talking about the body of the muscle. When we're talking about fascicles, we're talking about the next level of organization. So we're talking about these bundles of muscle fibers. So when we look at this, each one of these little circles within the body of the muscle is a fascicle. The muscle fibers, let me give you some arrows here to help a little bit. The muscle fibers is going to be when we're pulling these fascicles out, and then we're looking at these individual bundles within the fascicles. The muscle fibers are going to, again, be the cells of the muscles. These are going to be just long and fibrous, and they're going to contain our myofibrils. Our myofibrils are going to be the organelles within the muscle fibers that are going to contain proteins called myofilaments. And the myofilaments are the proteins that are responsible for that muscle contraction. So you can see from here, we're pulling out a myofibril, and then within that myofibril is when we get down to these little bitty tiny filaments, and those filaments all working together are what actually cause this whole muscle to contract overall. So what holds this all together? Thin sheets of connective tissue are going to enclose and stabilize the different layers of muscle and other tissues throughout the body. We refer to these thin sheets of connective tissues as fascia. Whenever these are cord-like, we call them tendons. Tendons connect muscle to bone. And whenever they're more like a sheet that lies over something, like you'll see this over like your abs and things like this, we call this an aponeurisis. So these are going to be sheet-like tendons. They're going to connect muscle and bone and other tissues, but they're just very long and not cord-like at all. So when we are looking at this muscle in general, whenever we see this layer that surrounds the whole big muscle on the outside, we call that the epimysium. If you think back to like your medical terminology, we know that epi means like upon, right? It's above, it's usually our top layer. Anytime you see my, that's going to be the abbreviation for muscle tissue. So epimysium is going to surround the whole tissue and lie over the top. Perimysium, when we see peri, we know that that means it's going around. That perimysium is going to cover each of those little fascicle bundles within the overall muscle. And then endomysium, right? Endo means inside. Endomysium is going to cover each individual muscle fiber. Here is, again, just another picture for you all to look at this a different way in case the other pictures were not working for you. And then here again is a breakdown of those words we just talked about. So anytime you see myo or mys, that's going to mean muscle. Sarco means flesh. It can also be used to refer to the muscle sometimes, depending on how it's used. Epi is going to be upon, peri is going to be around, and endo is going to mean inside. So let's take another look at this. We're just going to kind of keep zooming in and zooming in and zooming in a little bit more and a little bit more and a little bit more. So looking at skeletal muscle fibers, we know that they're multinucleated, they're striated, and they can be really long. There's going to be a few different organelles within these muscle cells. In the same way our regular cells have organelles, our muscle cells do too. They're going to have different names, but they're going to have most of the same functions. So try to just make these names match up in your head to what you already know about cells. The muscle cells are going to have a cell membrane. That cell membrane, when it's in a muscle cell, we're going to call a sarcolemma. The cytoplasm of the muscle cell, we're going to call sarcoplasm. And the endoplasmic reticulum, which is so important because it stores the calcium in our muscles, and we will talk a lot later on about how important that calcium is to those contractions. The endoplasmic reticulum is called the sarcoplasmic reticulum in muscle cells. We also have T-tubules. These are going to be folds in the cell membrane. They're going to help transmit signals into the cell. And then again, we talked about this already. We have myofibrils. These are our rod-like organelles, and these are going to contain those myofilaments, which are our contractile proteins. So the myofibrils are going to be specialized organelles. They're going to be tightly packed into a muscle fiber, and the proteins that they contain slide along one another to relax and contract the muscle cells. We refer to actin as the thin protein filament and myosin as the thick protein filament. The reason we call them thick and thin has to do with the banding. So if you look at these striated bands, you will notice that the dark bands are a lot wider than the light bands. So when we're talking about the light banding that is thinner, we're talking about actin, and when we're looking at those dark bands, we're looking at myosin. And here is another picture, just kind of again explaining that banding. Our actin we refer to as I bands. A good way to remember that is light or coloring with the I there in light. The myosin, which is our thick filament, is going to cause our A bands. And they have the darker coloring. So I gave you all this to hopefully help you remember this a little bit better when you're looking at these under the microscope. So the specific arrangement of the myofilaments in the myofibrils form contractile units, and we call these sarcomeres. These sarcomeres are going to be joined end to end within the myofibrils. So let's look at this a little bit more closely. So again, the sarcomere is going to be this contractile unit of the muscle fiber. There's going to be thousands in each muscle. They're going to be connected and they're going to be repeated kind of like little train cars. They're going to contain sections of actin and myosin proteins. These are going to pull on each other and cause muscle contraction. You can see that in this animation that I've put over here. Contraction is going to shrink the sarcomere. All the sarcomeres in a muscle fiber are going to contract at the same time to cause movement. So that's an important takeaway, that these things aren't happening incrementally. They're all kind of happening at the same time. Let's talk a little bit more about these bands. And what these bands explain is just how this looks under a microscope so that we can understand how the arrangement is. And then again, it's that whole structure causes function thing. Once we understand how they're arranged, the sliding filament theory and the stuff that comes next makes a lot more sense. So the striation of in skeletal muscle are going to be due to the arrangement of actin and myosin in the sarcomere. When we see those I bands, we know that that is actin. When we see the A bands, and right here is a really good thing, sorry, a little diagram, not a thing. Here's a good diagram that is showing you that. So like you can see, here's the A band. The A band is going to correspond with this. We can see the H zone. We can see the Z line, which goes up through here. So as you're looking at this diagram, compare it to this one, and it will help you to kind of see these different areas. Okay, so Z lines. Z lines are going to be the attachment of the actin proteins. So you can see here's our actin running through it, and you can see the Z lines are where the actins attach to the other actins. The H zone is going to be the area where only myosin is. So you can see in the H zone, all that you have here are just these purple myosin proteins. And the M line is going to be the middle of the sarcomere where the myosin is going to connect. So right along here, right? Okay. I know this is a little confusing, but once we get deeper into sliding filament theory, it will make sense why we're talking about all these things. Here is another picture for you all in case you need to see this differently. Okay, so let's talk a little bit about the steps leading up to this, and then we will talk about muscle contraction itself. So the real question is here is with these muscles, who's telling the muscle to move, right? So we have what we call a motor unit. A motor unit is going to be a motor neuron and all of the muscle fibers that it's going to signal. So the skeletal muscle will only contract when stimulated by that motor neuron. And each neuron is going to connect with a group of muscle fibers. So it's usually not just one, it's usually a group of them. And this is going to lead all of those muscle fibers in that unit to contract at the same time. Here's another view of this, just so that you're able to see. Here's our axon coming in, and then the axon terminal, which we call the neuromuscular junction, is coming right down onto that sarcolemma of the muscle cell there. Here's another view. We have a microscopic image of these axons branching and syncing up with these muscle fibers. And then here is just a breakdown of reminding us that with the spinal cord, we have afferent nerves and efferent nerves. Remember, those afferent nerves are carrying signals to the brain. The efferent nerves are carrying it away from the brain. So whenever we have the brain and we're sending a message to the muscle to tell it to contract, it's going from the spinal cord into those efferent nerves. And then it is coming down to that neuromuscular junction that is with on the muscle tissue. And that is how all of the anatomy of that kind of works. So that brings us to what we call the neuromuscular junction. This is just the synapse, or the meeting space, between a motor neuron and the muscle fiber. This is where the neurons kind of like talk to the muscle cells. It's going to require both calcium and acetylcholine to signal those muscles to contract. It is important to note that the neuron and the muscle fibers do not actually touch each other at the junction. That is an empty space, and those signals are sent using chemical messengers across that synapse instead. So, acetylcholine is very important in this process. It is a neurotransmitter, and it is used to send signals from a motor neuron to a muscle fiber that it's time to contract. I would definitely know this vocabulary word here. So here's how this process actually plays out. The first thing that happens is an action potential is going to arrive at the axon terminal. So you're going to get a little electrical signal saying, hey, I need this muscle to do something. In response to that, calcium channels are going to open, and calcium is going to flow into that axon terminal. The calcium entry is going to cause these little synaptic vesicles that we have here to release their contents. We talked about exocytosis when we talked about the cellular unit. This is what this is. We have these vesicles ready to do exocytosis. The calcium moves in, stimulates the vesicles to release their contents, and their contents are that acetylcholine, or that neurotransmitter. So the acetylcholine is then going to diffuse across that synaptic cleft, I got tongue-tied, and bind to the receptors in the sarcolemma. Once they have bound to those receptors in the sarcolemma, we will get a back and forth of sodium and potassium ions that is actually going to cause the muscle to contract. Sorry about that. My mouse slipped and I skipped ahead just a little bit on you all. So we will talk a little bit about sliding filament theory, and I'm going to give you the general steps. We talked a little bit about this in lab too. This is one of those concepts that's kind of hard to wrap your head around if you're just hearing someone talk about it or you just see it once or twice. There are some exclamation videos in this presentation, and I'm also going to drop a few of them in the study tools and resources, because this concept is, once you get it, but it is kind of hard to get over that initial hump and be like, oh, okay, that makes sense. So definitely don't feel like this is a thing that, oh, I'm going to read it in the textbook and I'm going to look at this one presentation and then I'm going to have it. This is something that you'll probably need to see a few times before it finally clicks. So what the sliding filament model is it is the theory of how these sarcomeres contract and relax. And at the most basic level, the contraction of a muscle is going to occur as the thin actin filaments slide past those thick myosin filaments so that they overlap and cause something we call cross bridges. So this is a very, very, very basic overview of this process. I really do want you to watch some of those animations over and over again to really get this. So the first step in this process, we're going to have that action potential. It's going to move down the neuron, and it's going to allow those calcium channels to open and enter the neuron, pop those vesicles, and then release that acetylcholine. ACTH, or ACH here stands for acetylcholine. That acetylcholine is going to bind with the receptors on the muscle fiber. That is going to cause the action potential to continue into the muscle fiber. That action potential is going to travel down the sarcolemma along those T tubules, and eventually it's going to stimulate the release of calcium from the sarcoplasmic reticulum. The calcium at that point is going to bind to troponin. It's going to change shape, and those myosin sites are going to be exposed to the actin. At this point, the myosin is going to form a cross bridge with the actin. The myosin heads are going to kind of pivot and pull on the actin filament toward that M line, and it's going to pull it in. ATP is going to attach to myosin, and at that point, the cross bridge will detach, and then myosin is reset and ready to be used again when stimulated. So again, this is the very, very most basic set of steps broken down into like one sentence at a time, the smallest that I could break it down. But this is very, very simplified, and to really get the concept, really, really tune in and watch those videos. So let's talk a little bit about just things that can go wrong and how to know what's happening. So muscle cramps, these are going to be our sudden or involuntary contractions of muscles. The causes of this can be due to long periods of exercise, physical labor, some medication, dehydration, muscle strain, all kinds of things can cause this. Mineral depletion, if you get low on calcium or potassium or magnesium, you can have these muscle cramps. We can treat this with stretching and exercises and things like that, or applying cold and heat. Let's see, there's another one. Let's see, another thing that your book talks about briefly that I don't want you guys to get too stuck on, but I want you to understand how it works, is creatinine. If you've ever been really working out or bodybuilding or whatever, you're probably familiar with creatinine. The reason that creatinine works is because it increases the regeneration of ATP. Remember, that's our energy molecule. And it increases the regeneration of this for those sliding filaments. So that is how that works. Let's talk a little bit about the different types of muscle fibers. So you will have slow twitch muscle fibers. We call that type 1. These are going to be able to do a slow contraction and have a very low force, but are going to have a pretty high endurance. You also have fast twitch muscles. These are going to be able to have a moderate contraction, so you can get a medium force for this, but they're going to have a little bit lower endurance. And then your other type of fast twitch, which is your type 2B, is going to be a fast contraction, a high force, and very, very low endurance. So this is going to be the difference between like long distance running and being able to like just run a relay race really fast with these muscles. Here is another review video of the neuromuscular junction and sliding filament for you guys. I will post a few of these because again, it is kind of a complicated concept. And let's talk a little bit about body movements. So body movement is going to be attained due to the muscle pulling on an attached bone by moving that bone at the joint. Muscles are always going to pull, they're never going to push. Muscles are going to be attached to bones at least 2 points. The origin is going to be the attachment to an anchoring bone, and the insertion is going to be attachment to a movable bone. We're going to have a few different types of angular movement that your book is going to talk about. Your book actually has way more than these, but I just want to talk about a few of them. So we have flexion, which is going to decrease the angle of the joint and bring the two bones closer together. We have extension, that's going to increase the angle of a joint, and it's going to be the opposite of flexion. Dorsiflexion is going to be when we flex the foot closer to the shin. Plantar flexion is going to be when we flex the foot further from the shin. And hyperextension is extension beyond the normal range of motion. let's talk about these two. We also have abduction. Note the ABD there. This is going to be moving the limb away from the midline. And we have adduction. This is going to be moving the limb towards the midline. So think about this like adding your arms to your middle is an easy way to remember that one. We have circumduction, which circumduction is going to be the circular movement of the distal end. Rotation is going to be movement of a bone around an axis. So this is like shaking your head no. Pronation is going to be turning inward. So this is like your palms facing down. Supination is going to be to turn outward. This is going to be when your palms are facing up. And eversion is usually referred to with the foot, and it is that ability to kind of turn the foot inward, whereas inversion is turning the foot outward. Opposition is going to be the movement of the thumb towards the other finger, where retraction is going to be moving a part backward. Protraction is going to be movement of a part forward. Elevation is lifting a part in the superior direction, while depression is going to be lowering a part in the inferior direction. These can be really helpful once you actually are getting into medicine and moving people around and that kind of thing. Let's talk a little bit about diseases, disorders, and other changes in the muscular system. We're not going to talk a whole lot about this. I just want to touch on it a little bit. So you will see a word, the word hypertrophy. And what hypertrophy is when muscle fibers are enlarged. This is through microscopic tearing and repairing of those myosin and actin proteins. So we see this with like bodybuilders when their muscles get really, really big. We can also have atrophy of muscles. This is when the muscle fibers actually shrink and weaken. This can be caused sometimes due to disease. This can also be caused just to like not using the muscle. And when the muscle is not used, the proteins inside that actin and myosin are going to be broken down. Myasthenia gravis, I want to talk a little bit about because we talked a little bit about this in lab. This is an autoimmune disease that leads to progressive weakness due to a loss of those acetylcholine receptors on the muscle fiber. So when that acetylcholine can't bind, when any of those steps at the neuromuscular junction don't happen, none of the steps following it can happen either. So this is going to lead to progressive skeletal muscle weakness. And you can have like face paralysis and things like that. And let's see, in kind of the same vein, botulism is usually a rare and potentially fatal disease caused by the botulism toxin. But we also use this like in Botox. So what this does is it prevents contraction and causes smoothing of some muscles because it actually is blocking that calcium reuptake at the neuromuscular junction. So that's all that I have on disorders. Let's talk a little bit about how these skeletal muscles are named, because we're going to get a little bit into specifics on skeletal muscles, but if you know how they're named, that definitely will take you pretty far in being able to figure out at least a general area of where they're located at. Okay, so the first thing we're going to look at when we're naming skeletal muscles is usually the location of the muscle. And as an example, many muscles are going to be named for the bones that they attach to, right? Like the temporalis is going to be attached to the temporal bone. So if you've already taken the time to memorize a lot of these for the skeletal system, you're going to probably already be halfway there. Also, the number of origins. So like on the triceps, we have 3 different origin points. There's like 3 heads to that muscle. So you can use that to get you pretty close to knowing the name of that muscle. And then the location of the insertion as well. So like, for example, anything that says sterno is going to have either an origin or an insertion on the sternum. The shape of the muscle can be really helpful. There's several different muscle shapes that your book goes over. Like, for instance, the deltoid is called that because it is triangular, right? The trapezius is called that because it is trapezoid shaped. So that is definitely something that can be helpful. And then the action of the muscle. So you will see things that say like flexor or extensor. So based on what the muscle is actually doing, what it's actually moving, can help you with the naming. Okay, so this is pretty much all that I have. I have included a whole lot of pictures for you because when it comes to learning the names of these muscles, unfortunately, this is kind of a memorization thing. Like learning where they are, learning what they do. I tend to really like things that are more process based, where you're like learning a concept, but in anatomy and physiology, sometimes for the anatomy part, it is just about learning where it is and what it does. I included some different pictures for you all here on all of these slides at the end. These are also in your book, so I just again wanted you all to have a few different views of this to be able to look at it and remember the names of some of these. I will say there are quite a lot of labels on these images. I really want you to focus on just what your book talks about, and I love the way that your book does this. So if you go to page 205 in your book, it is going to give you like an overview of each of the most important muscles. Those are the only ones I really care about you knowing. And then if you continue reading, whenever you get down to page 211, 212, sometimes it's a little bit hard to tell on the ebook because it crosses over like right at this point, there is a table. And first there are figures like the ones that I've shown you here, where it's going to show you where all of those muscles are located, like with a labeled figure. But also there is a table at the very bottom that just says, here's the name of the muscle, here's its function, here's what it does. That is very, very good stuff to know. So essentially like page 205 to 211 is all then broken down in that one table. And it's going to break it down into very specific sections of the body. And I would spend a whole lot of time there. If you have any question, if you aren't quite understanding what a specific muscle does, we will talk more about that in lab, but we can definitely sit down and do some like demos and whatever with our own bodies. So that is an option, but yes, I want you to look at that. I also included 2 different crash course videos at the end of this. I like crash course because it gives you kind of a good intro and overview. It is absolutely not every single thing that you need to know, but they do a really good job of explaining the basic intro stuff. So I included those for you as well. There will also be lots of stuff in the resources folder for you all, and as always, I'm here. So even in between labs or whatever, you can always reach out to me if you have any questions, concerns, anything at all. If it's just not sticking, let me know, and I'm happy to help.